immunoglobulin gene rearrangement, I've kind of alluded to that in one of the previous clips for this chapter, but I think it was chapter 5 also that covered in quite a bit of detail. So you get a lot of genetic rearrangement in there, and as to my analogy before of using a table, they're going to build one leg, so they're going to build a heavy chain gene rearrangement. And fortunately for humans, we have two alleles for the same set of characteristics. So if some error crops up in one chain, then it can shut down that pathway and open up another pathway on the other allele, which gives you two chances at producing a heavy chain for each of the heavy chains and for each of the light chains. What has to happen is you get, of course, the VDJ genetic rearrangement that can occur in the heavy chains. And when the heavy chain is completely gone through its rearrangement, it can be kind of classified as a pre-B cell. Now, they do use well, I won't say in some cases, in all cases, there's kind of an intermediate little protein that binds to the receptors to the primitive immunoglobulins with just the heavy chains that serve as temporary light chains. What did they call them? Surrogates, or yes, surrogate light chains of pre-B cell receptors. Then you get all of that RAG1 and 2 and the other genetic rearranging systems that allow you to get your DNA to twist and bind together in different genetic sequences. Now, these immature B cells express a membrane-bound immunoglobulin M, IgM, which is a monomer. That's an important fact. It's a monomer at this point. It's not until it gets activated much later on that it would serve to produce the pentamer form of the MIgM. And some of them also have the membrane-bound immunoglobulin D on their surface, but we really still don't know what the function of that is. Mainly because they have taken mice and they've done some knockout experiments where they've knocked the IgD out, and yet they still produce about the same number of mature B cells as their normal wild-type cousins. I just think that's interesting, and we still don't know exactly what the IgD is doing. Once you complete reproduction of light chain genetic rearrangements, then that now commits this immature B cell to a very specific antigenic path. It can only recognize one specific antigen. But at early stages of development, before it has moved out of the B cells, before it has gone through, I guess you would say, a maturation process, if it binds to an antigen, then it will result in death, not a multiplication of that particular cell. I think we'll talk about that more in the next unit.